This video will include images of human remains. This is for educational purposes. If you're interested in this content without these images, I am considering a podcast option when I have time to record episodes and set up a place for those episodes to live. Within archaeology and biological anthropology, my research falls within the subdiscipline of paleopathology. So, what is paleopathology, and how do we do this research? Paleopathology is the study of disease in past populations. This includes infectious disease, metabolic disease, neoplasms, and malnutrition and stress. It can also include degenerative joint disease like osteoarthritis. While a lot of this work starts with the human skeleton, the goal of paleopathology is more than simply making a diagnosis for any single individual. Paleopathology brings together archaeology, biological anthropology, medicine, and genetics to try to understand the connections between disease and human behavior. A quick note here. When I use the prefix paleo here for paleopathology, I don't mean that we're only looking at the time period referred to as the Paleolithic, the Stone Age. Paleo means ancient or old and comes from the Greek word for ancient or old, paleos. Sorry for my Greek pronunciation here. I took Latin and not ancient Greek. We use paleo to mean the past generally, not just time periods that we would consider. Paleopathology has several means of investigating human remains, most of which involve slapping the prefix paleo onto the names of other scientific disciplines to emphasize the fact that the context of this research is the past. We're often working in the context of the archaeological record. The study of skeletal trauma is often included within paleopathology as well, though I think that deserves its own video. For the purposes of this video, I'm going to focus on disease because that's my specialty. Once skeletons are excavated from an archaeological site, how do we get started with paleopathological analysis? What are our sources of data? We usually start with macroscopic analysis, which is a fancy way of saying we look at the skeleton for evidence of disease. I used my eyes and recorded what I observed. Okay, but what are we looking for in a skeleton that might indicate some kind of disease process? We're usually looking for evidence of bone acting in unusual ways before an individual died. This could be bone destruction, what we can refer to as lytic lesions or osteoclastic activity. Osteoclasts are cells that break down bone. A lesion is part of an organ or tissue that has been damaged through an injury or disease. Lesions in different parts of the skeleton can be suggestive of different disease processes. Lesions on the visceral surfaces of the ribs, so the sides facing the viscera, your internal organs, can suggest a chronic pulmonary condition. Destruction of bone in the spine could suggest an infectious disease like tuberculosis or brucellosis. We can also look for the unusual buildup of bone remodeling, or osteoblastic activity. Osteoblasts are cells that build bone. This could be bones fusing together that wouldn't normally do that, or something like periostitis, the chronic inflammation of the periosteum, which is the layer of connective tissue that surrounds your bones. Excessive bone buildup in the form of tumors gives us paleooncology, the study of cancer in past populations. Some disease processes, such as those caused by tuberculosis, can involve both bone destruction and bone remodeling. Depending on what we're looking for and what equipment we have access to, we can do paleoradiology. Paleoradiology is a fancy way of saying X-rays and computer tomography, CT or CAT scans, but paleo. You'll also see this referred to as paleoimaging. Once again, we have two names for essentially the same thing for reasons I cannot fathom. Paleoimaging has become especially important for studying mummies, because X-rays and CT scans are a non-invasive way of collecting data. The preservation of soft tissue in mummies means that we can find evidence of diseases that may not be available with a skeleton, like calcified lesions found in the lungs of the individuals from the Watts mummy collection who had tuberculosis infections. Mummies can also help us engage in paleohistology, the study of microscopic structures of preserved tissues, and paleoparasitology, the analysis of parasites in the past through finding preserved eggs, larvae, or whole-ass worms. While we can find evidence of parasites in the preserved intestines of a mummified individual, we don't necessarily always need a human body to do some paleoparasitology. Sources of data for these studies can include soil sediments and coprolites. Coprolite is the fancy word for fossilized feces, and that kind of work warrants the use of bigger pipette tips than what I use, and I'm glad it's not my job. No joke, I think the box of pipette tips that I found in our ancient DNA lab is labeled, telling me that they're for poo. So, what do you do when you have human remains with indicators of a disease process, but you're not sure which disease? This is when we do what's called differential diagnosis, because some diseases will present similar features when they affect bone. Most infectious diseases will not affect bone, so are you looking at a metabolic disease like scurvy? Part of differential diagnosis is ruling out potential diagnoses and explaining why you ruled it out. So what can you do to rule out diseases and support a particular diagnosis? Beyond the methods we've already discussed and a thorough examination of the remains and comparison to what has been published about these different diseases in both paleopathological and clinical literature. If you're looking for more evidence of infectious disease, you can do paleomicrobiology. This does depend on being able to take certain types of samples from the remains, having access to labs that can run these kinds of tests, having people on your team who know how to do the lab work and how to analyze the data and interpret the results, and being able to pay for all this. Whole genome shotgun sequencing is not cheap. While this can include chemical tests for biomarkers that are unique to specific diseases, I'm going to focus on DNA analysis here because that's what I do, and it's what makes me the most excited to talk about. We used to test DNA samples from human remains and target either one specific disease or multiple diseases by looking for a gene that's unique to the causative agent of that disease. If we found that gene in the sample, that individual was likely infected with that disease at the time of their death. It doesn't necessarily mean that they died from that disease. They could have had comorbidities, and we might not find the genes we're looking for all the time because it just might not be preserved. That's just part of the nature and the limitations of working with fragmented ancient DNA. But studies like that helped us figure out that the Black Death in Europe was caused by the disease plague, whose causative agent is the bacterium Yersinia pestis. I have spent 4 years of my life reading plague DNA studies, but you know what? No regrets. Excuse to make cute plague doctor plushies, what more could I want? I didn't need an excuse. The diseases chosen as the targets for studies, like all of those plague studies, aren't always based on what we can observe in human remains. Remember, infectious diseases, most of them will not actually change what your bones look like. Diseases like plague or epidemic typhus don't change bone, so DNA analysis allows us to choose diseases based on geographical context and time period. However, DNA sequencing technology has come a long way since we started doing paleomicrobiology in the 1990s. Now we can sequence whole genomes from samples in a way that won't exclude whatever's in those samples. We can sequence the DNA, grab whatever is in there, and figure out what that DNA belongs to, if it's human, bacterial, viral, or even fungal. This is what I do when I release the Kraken. Kraken 2 is the program I use to identify different species, and I will never get over how cool the name is. Quick little side note here, I don't refer to myself as a paleomicrobiologist or generally describe what I do as paleomicrobiology. Personally, I find it too clunky and too specific, and my training started in osteology and skeletal analysis. I'm not a geneticist or a microbiologist. The most specific I get is saying I'm a paleopathologist and calling it good. Once we have all of this data collected and analyzed from individuals, we can do the last paleo on our list, paleoepidemiology. How do diseases and other health conditions affect populations? But paleo, because they're past populations. Ancient DNA has been particularly useful for this if we look at an example like all of those plague studies. The more evidence of plague infections that we find in the archaeological record, the better understanding we have of when and where outbreaks happened in the past. By comparing those genomes, we can see which strains were causing those outbreaks and how they spread through different populations. There's a lot more to paleopathology than what I've covered here, but I'm hoping this video has served as an overview of the discipline, if a rather biased one. There are entire textbooks for paleopathology. I use Ortner's identification of pathological conditions in human skeletal remains. Let me know what you'd like to know more about, and I'll see you next time. Thanks for watching.